characterizing dynamics in electrophysiological data. She is also a very generous mentor and has been a resident faculty at the Woods Hole course for methods in computational neuroscience ever since 1999. I don't know how many of you were in neuroscience at that time. With that, let's welcome Sara. As you're setting up your screen sharing, I will go and remind people of a little bit of housekeeping. This is a live feed that is also being recorded, and it will be broadcasted on YouTube afterwards. If you have questions, please use the Ask a Question tab in the bottom of the screen, so the rest of the audience can upvote the questions based on interest. I will interrupt Sara during the talk to ask your questions. So with that, take it away.
Okay, thank you so much. Thank you so much for the invitation. It's a real pleasure. Your center is a fantastic place, and I'm just honored to visit and to have the chance to actually close your academic year. So let me share my screen. And there we go. So what I want to talk to you about today is really based on my view that as the important pathway to understand how the brain works is to really look at populations of neurons. It's of course very important to do single neuron work. It's very important to understand how single neurons react to their inputs and produce action potentials. But if we want to study the brain and understand the brain as an organ that is capable of processing information, we need to deal with the question of neurons working together in order to process input, in order to make decisions, in order to produce an output that results in a motor output. And so to study the populations and to study the dynamics of these populations becomes very important and becomes a necessary component, I believe, on our quest for understanding how the brain works. So what I'm going to do is I'm going to tell you about, I'm going to focus on motor cortex, which is what triggered this kind of research that I will be sharing with you today. I'll start telling you about a simple motor task that I will use as a kind of a prototype or an example during my presentation. And then I'll tell you about this concept of manifolds, of neural manifolds, and how they relate to the control of movement. And then I'll tell you about how far we can go by pushing a linear approach to the analysis of neural activity. So the work that I'm going to present has been done in collaboration with Lee Miller, who is also a professor at Northwestern, and he runs a monkey lab, and Lee and I have collaborated for many years now. It's a very gratifying collaboration. I think every theorist should have an experimentalist that he or she really loves to collaborate with, somebody that you trust, somebody that trusts you, and somebody that is open to a real interplay between theory and experiment. I am myself trained as a physicist, where the interplay between theory and experiment is a long established tradition, and I am very happy to see that neuroscience is also moving in that direction, that as the methods for acquiring experimental data become very sophisticated, and both the nature and the quantity of data change in a qualitative manner, the interaction with theory and the idea of doing data analysis that is model-based has become more and more necessary, and that has triggered a very interesting collaboration between theory and experiment, which I think has allowed us to make a lot of progress and will allow us to make even more progress in the future. I want to mention Juan Gallego, who was a postdoc with Lee and myself at the time this work was done. He's now an assistant professor at Imperial College London, where he has set up his own lab. And Matthew Perridge and Raid Charpuri were both graduate students at the time this work was done. Raid is now doing a postdoc at Pitt with a group of Aidan Batista, and Matt Perich is working with Canonical Correlation in New York at Mount Sinai. Okay, so let's start with a simple motor task. So imagine that you are a monkey presented, or a person, a human, presented with the following task. You have a center target surrounded by 8 targets arranged in a circle. You are focusing on the center target via, be it mechanical, by touching it, or by placing a cursor on the center target through the use of a manipulandum, as is shown here. One of the 8 peripheral targets is lit. That will be your target for that particular trial. You have to wait. It's a delayed, it's an instructed delay, and the delay period changes randomly from trial to trial. It's between 500 and 1,000 milliseconds. Then you get the go cue. When you get the go cue, you're supposed to start a motion, a reaching motion that lasts of the order of 300 milliseconds between the go cue and the movement onset. The movement onset then is followed by a trajectory that allows you to reach the desired target, and you stay there, you get a reward. Then you get back to center, and then your trial comes in. So it's a very simple reach task with only 8 possible targets, where the modulation are the direction theta of the target and the reach of the target, which is just fairly well established. You need to be within the target, but there is some allowance for variability within that. So, oops. There. The story of this task, or the neural investigation, or the correlation of the neural correlates of this task, starts in 1982 in the lab of Georgopoulos. At that time, people could only do single neuron recordings, so he implanted an electrode and recorded the activity of a neuron in primary motor cortex, in the arm area of primary motor cortex, so that this neuron was, the activity of this neuron was related to the activity. And he recorded many trials, in this case, every row in this representation corresponds to a different trial. The ticks, of course, are the spikes emitted by the neuron. And what you see is that, for instance, if the reach was in the downwards direction, there is almost no variability in the activity of the neuron. 0 is the go cue, and then we are going both 500 milliseconds before and 500 milliseconds after the go cue. So focus on this 1 second of activity. And what you see is that there are directions in which the activity of that neuron is actually suppressed. All the movements that imply going to the left show a suppression of activity, while the directions, while the reaches in, I'm sorry, that was going to the right, while the reaches going to the left show an increase in the neural activity. So there are 2 things to notice here. First of all, there is a lot of trial-to-trial variability. Every row, even in a given direction, every row is different from the other rows. But at the same time, there is a specificity that this neuron enhances its activity in reaches that go to the left and decreases its activity in reaches that go to the right. So based on the recording of many neurons like this, Georgopoulos and his group proposed in 1982 what they call a tuning curve model, a cosine tuning curve model for these cortical neurons in the primary motor cortex, such that if the reach was a direction theta and for an extension r, the firing rate of this neuron I, would consist of a background bi, which is the minimum firing rate of the neuron, and a cosine modulated curve centered at a preferred activity theta I. So the main direction dependent term, the only direction dependent term here, is cosine of theta minus theta I. The cosine, of course, goes between -1 and 1. So we add 1 to it. Now it goes between 0 and 2. And we get 1, we divide it by 2. So now we get a modulation that goes between 0 and 1. A sub I is the amplitude of that modulation. It's the extent between the minimum and the maximum firing rate, the difference between the minimum and the maximum firing rate of that neuron. The maximum firing rate specifies or determines the preferred direction. And the extent r of the reach, the distance that needs to be travel in order to reach the target is also a modulating factor in this very simple model. So the way we do the experiment now is with multi-electrode arrays that allow us to measure many neurons at the same time. So this is the typical electrode array used in the Milan lab. It's 4 millimeters by 4 millimeters. It's 100 electrodes. So distance between the adjacent electrodes is 400 microns. And we implant this array in primary motor cortex. As you know, primary motor cortex is ear to ear, essentially. And there is a homunculus in primary motor cortex that tells us where is the arm area. And the way we know that is because if we put an electrode there and inject a current, we will see a twitch. So we go to the arm area of primary motor cortex, and that's where we implant the electrode array contralaterally, of course, to the direction of the ridges. And now we get data that looks like this. This is the current status of the data that we get in the Milan lab. We can record of the order of 80 neurons or 100 neurons. This is about 20 seconds of data. Every tick, again, is an action potential. It's a spike emitted by a neuron. But the difference now is that every row is not a different trial. This is a long trial, 20 seconds of data. I'll show you in a minute that during this trial, there were 3 reaches to 3 different targets during this length of recording. And now every row represents not a different trial, but a different neuron that is being recorded simultaneously. So as I said before, during this about 20 seconds of recording, we have seen the monkey executing targets, executing reaches to 3 different targets. So the yellow target, and then the red target, and then the blue target. I'm going to use a color code for the different targets. So what we're going to do, first of all, we're not going to study in a very simple manner, not concerning ourselves so much with the dynamics of the reach, but to the global activity during the given reach. So in order to do that, we select a window, a very wide window, 300 milliseconds, and we place that window at the onset of motion, around the onset of motion, as this is target presentation. There is, as always, a variable delay period between them. And for each one of the neurons, then we can measure the firing rate corresponding to that particular reach by simply measuring the number of spikes that we observe during this interval and dividing that by 300 milliseconds, we get the firing rate. So now I have a firing rate for 80 neurons or 85 neurons measured simultaneously. And so if I wanted to visualize this activity, I would need to go to a neural space where every axis represents the activity of a neuron. So that would have to be an 85-dimensional neural space. Well, that's very hard to visualize. So in order to show you the data, I'm going to pick 3 particular neurons. Neuron 46, which is a neuron that fires a lot. Neuron 70, which is a neuron that fires very little, and neuron 78, which fires kind of in an irregular manner that might be interesting. And the question that I want to ask is the following. If I know the firing rates of these neurons, can I tell to which one of the targets the corresponding reach is going to go? So can I decode the neural signal in this very, very simple manner, just 1 out of 8 targets? Can I say that this point of neural activity in the full neural space corresponded to yellow target, corresponded to the red target, or corresponded to the blue target? So let's look at this 3-dimensional representation that is totally arbitrary. I just pick 3 neurons at random. So again, the targets are color code. Every trial, every reach is represented by a single point. It's a single point in an 85-dimensional space, but I'm showing you here that point in a 3-dimensional space corresponding to the firing rate of neuron 46, neuron 70, neuron 78. So every point here corresponds to 1 trial. And after I put all the points in this space, there is no dynamics. This is just statistics. 1 point per trial, and after I put all the points in this space, I color code them according to the trial. So now I ask myself, can I tell from the location of this point in this low dimensional representation of the neural space, can I tell in which direction the target, the monkey was going to move its arm? Can I tell which one was the target corresponding to this particular trial? And you can look at this data and immediately tell me that I'm a fool if I expect to be able to do that. Why? Because there is no clustering here. There is no clustering points corresponding to the same target are not clustered together. They are not separated from points corresponding to a different target. So there is no way for me to know from the location of the point. Look at the point here, going to red target. Its neighbors are orange and green and blue. So the location of this point cannot be enough to tell me in which direction the reach is going to happen. That is to say, in which direction, which is the target that corresponds to that particular trial. So this was hopeless. And then what did we do? We went back to good old Georgopoulos and we said, well, we know that there is this hypothesis of a cosine tuning for direction of motion, in which the cosine curve, just using 3 parameters, a baseline, a preferred direction, a baseline, and an amplitude of modulation, can tell me what is, so within the simple model, what is going to be the firing rate of a given neuron, given these 3 parameters for a neuron. So I said, well, I'm a physicist, so the simplest approach to this is to think of a system of non-interacting neurons, which is totally ridiculous as a model, because these neurons are interacting with each other, they are part of a network, they are interconnected. But let me think, as a first approximation, a system of non-interacting neurons, and these non-interacting neurons, each one of them is born with the tuning curve, with the cosine tuning curve, according to Georgopoulos. So there is acting in this particular task as if all its activity was controlled by this tuning curve and nothing to do with what other neurons are doing, except that, of course, they will be correlated through the direction of motion, but they are otherwise conditionally independent. So what will happen if I now have a population of such neurons, non-interacting, each one of them cosine tuned, and I make a reach in a direction theta and of extent r? What would be the firing rate of the population? So this firing rate now is an array of n numbers, one corresponding to the firing rate of each neuron. And it's just a simple page of algebra to convince yourselves that this firing rate will be described as follows. There's going to be a vector B, which is the collection of the n baselines, one for each neuron. Then I'm going to have the same 1/2 of r, the vector A, which is the vector of the amplitudes of modulation for each neuron, the difference between the maximum firing rate and the minimum firing rate. And then I get a linear combination of 2 vectors, phi x and phi y. Phi x and phi y are 2 n-dimensional vectors. The components of phi x are of the form amplitude times the cosine of the preferred direction, and the components of phi y are of the form amplitude times the sine of the preferred direction. So these 2 vectors, phi x and phi y, are just 2 vectors in this n-dimensional neural space. These 2 vectors are not necessarily orthogonal and they don't have length 1, but they are linearly independent. So these 2 vectors span a 2-dimensional hyperplane that is located somewhere in this n-dimensional space. And in this 2-dimensional hyperplane, as I vary theta, I will trace some kind of an ellipsoid, because I am just simply varying the coefficients of the linear combination of phi x and phi y that I'm using. Then, of course, there is a displacement from the origin given by the vector B and the vector 1/2 of r times A. But this 1/2 of r is also modulating the linear combination. So now if I allow for slight variations in r, I will not get only 2 dimensions, but I will get a 3rd dimension, a smaller 3rd dimension that will allow me to predict what will be the firing rates of these neurons. So as you see, as we vary theta, we span some kind of an ellipse, a circular kind of shape that is contained in a 2-dimensional plane, and then I have fluctuations along a 3rd dimension that is due to the presence of this coefficient 1/2 r. So, and the direction will be, of course, the direction of the vector A, the direction in these fluctuations occur. So this gave us the idea that there is indeed a dimensionality reduction in this space, but that the way to look for it is not to pick 3 directions at random, it's not to pick 3 axes that correspond to 3 arbitrarily chosen neurons, but to pick 3 axes that correspond to A, phi x, and phi y. And the way to do it empirically is to simply take all your data in this very high-dimensional neural space and do a dimensionality reduction. And this is essentially what was done by the group of Krishna Shenoy at Stanford, and we were scooped by this. This was, the late aughts and almost in 2010. We have obtained a picture like this using principal component analysis. They did it using factors analysis. But the main point is that they found 3 directions in this space, and these directions I'm going to call neural modes, and we'll come back to that in a minute. And in this 3-dimensional representation, you can see that now we are in business because the points are clustered corresponding to different targets, and the clustering is fairly well separated. But moreover, what was really very surprising both to us and to Krishna Shenoy and collaborators was that these clusters are organized in this totally abstract representation in a manner that reproduces organizations of the targets in real space. You can see I go from red to orange to yellow to green. I go from red to orange to yellow to green. So the organization of these blobs of points in this space responds or reproduces the organization of the targets in the actual physical space, which is outside of the monkey and outside of the brain. So what are these neural modes? What are these axes? These axes are directions in neural space. Each one of them corresponds not to a given neuron, as in this very simple crude attempt that I showed you before, but corresponds to directions, to specific patterns of population activity. A direction has a different loading, a different projection along each one of the axes corresponding to different neurons, and it tells you that when a given neuron is active in a certain amount, what is going to be proportionally the activity of all the other neurons. So here we have 3 neural modes that suffice to give us a good representation of the activity corresponding to this particular task. In this very, again, it's a very simplified task, and I've taken a very simplified approach to analyzing the data, because what I'm doing is just giving you 1 data point per trial. I have chosen a delta t, a bin, in which to do spike counting, so that I capture most of the activity corresponding to the trajectory of the trial on the way to the target, and therefore I have captured none of the dynamics corresponding to actually going to the target and coming back. But we already see a low-dimensional representation, which takes us to this idea of neural manifolds that are task associated, that are task specific, and that allow us to explain what is the population neural activity. So let's go back to the data that we are taking. So in principle, what I would love to do, of course, is to be able to record the activity of all the neurons that participate in this simple reaching task. For a reach movement like the one that I am using here as an example to describe the basic ideas, there's of the order of 1 million neurons in primary motor cortex that will be modulated by the simple reach to 8 targets task. So that's what I mean by n infinity. The matrix, the ideal matrix, if I could capture the full neural space, would contain an infinite number of neurons, meaning, of course, not infinite, but would contain the activity of all the neurons that participate in that task. And as in the raster that I showed you, every neuron corresponds to a given row in this data matrix. And then in order to describe the time evolution of the activity, we pick a bin size delta, and we measure time in discrete iterations of delta. So we start at time t, then we go t plus delta, t plus 2 delta, as we get to t plus capital T, where capital T is the duration of the experiment in units of this bin size delta. And so now the columns correspond to different times. And the duration of the experiment is such that it involves many reaches to many different targets. Now, this would be the ideal case in which we have access to all the units that participate in this particular task. But that's not what we really do. What we do is we have an empirical neural space that is defined by the number of neurons that we can monitor with our electrode array. That would be of the order of 100. With neuropixels, it would be of the order of 1,000. But that's what we call the ambient dimension D, is the number of recorded neurons. For instance, in this case, this is the network of neurons in M1 that is participating in the task. And I have put 3 electrodes, N1, N2, and 3, and I measure the spikes emitted by each one of these neurons. So this is, yes?
Sara? We have a couple of questions that maybe would be a good time to take them before we move to the next analysis. So I'll start with a quick clarification question from Sean. He was wondering before if the 3 neural modes are all needed, in the example before, are all needed for the decoding, or strictly speaking, you only need a 2-dimensional space for the decoding.
That's a very good question. You can't get away with 2 dimensions. Actually, when we are doing the principal component analysis of this data, we were using 2 dimensions and not 3.
Okay, great. And we have a more complicated question from Yuri Buzhavsky. I'll try to read it all. So, Sara, the problem I have with many of the population analysis and dimensionality reduction approaches is that the contribution of individual neurons to the population patterns is strongly skewed, with a small fraction of neurons contributing disproportionately. Are there ways to incorporate this prominent inequality into
actually,
into this theory?
That's not quite true. Let me show you. Let me go back to my. So let me show you this plot. This is a plot of the loading. So each column here represents an eigenvector. And if I do principal component analysis, this is 2 different tasks. And this is a recording that involves of the order of 70 neurons. So in each column, you can see how many neurons are involved in that particular eigenvector. And as you see, it's a very distributed representation for most of the leading eigenvectors. And only when you get to the eigenvectors that are associated with a very small percentage of the variance, you begin to see what you call this biased loading, in which, for instance, a given eigenvector will involve maybe, for instance, let's look at the last one. It involves 2 of the neurons and not more than that. But that's not the case for the leading eigenvectors, the ones that account for most of the variance, which are the ones that we keep in our representation. As you can see here, these neural directions that are the neural modes are very, and now I have to remember where I was before.
Right. We were swamped by more questions, and we needed a break for people to unleash their curiosities. So a very quick one: do we know if the target direction selectivity is related to hand movement or eye movement, since they're strongly correlated?
Yeah, this is it's not hand movement, it's arm movement, actually. We are looking at the arm area of the primary motor cortex. And of course, they are correlated, but you can separate them temporally because, of course, the visual information comes with the activity, is activity that you see during the preparation period until you get the go signal. So that's based on vision, you could argue, because it's a vision that has determined or instructed through premotor cortex, through PMD, premotor dorsal cortex, which is the direction in which the reach is going to proceed. But only when you get the go cue and movement onset, you can talk about neural activity that is related to the actual motion, to the actual displacement of the arm. So because of this temporal difference, we can separate the two.
A very last one, and then we can move on. Sean is asking to which degree these representations are task specific. So is it about arm in a movement in a particular direction orthogonal of what you're trying to do, or if you change the task slightly with the manifold shift somehow?
It's an excellent question, and we don't know for sure, but I'll tell you what's my current view. My current view is that these manifolds are very low dimensional and they are task specific, and that in the very large neural space that is all of primary motor cortex, you have many of these manifolds that correspond to the tasks that you have learned. And every time you learn the task, you establish a manifold for that task. When you practice a task, and I'll talk about that, we'll go back to this in the last part of my talk, when you practice, if you're an athlete or a musician and you keep on repeating a given motion, what you do is you stabilize that manifold. And this is why manifolds associated with learned tasks are very stable, is because of the constant practicing. People talk about a muscle memory, but I don't know what that means because I don't know how muscles have memory, but the neural system has memory, and that memory is embedded in the interconnectivity of that particular network. And that I will talk about that as we go along, is associated with the establishing these manifolds. So they are in my view, they are certainly task specific. You could also think that there is a big mother manifold in motor cortex, and different tasks are associated with looking at different regions in that manifold. It's that possibility cannot at this point be discarded.
Mm-hmm. Great, thanks so much. I think we're ready to go on.
Okay, you interrupt me. You interrupt me again when needed. You stop me when there are questions. Okay, so this is what we call then the ambient dimension, which is an empirical neural space. It's limited by our recording tools. It's limited by what we have access to. And so now the data matrix is a little bit different. It is again the same number of columns because that's the duration of the experiment, but the number of rows is capital D, where capital D is what we call the ambient dimension. It's simply the number of neurons that we can record from, and as I said earlier, is of the order of 100 if you use these multi-electrode arrays that I showed you earlier, and it's of the order of 1,000 if we are using, or 5,000 if you're using neuropixels. And now it might become even larger if we think that calcium imaging can really distinguish between individual neurons. So this is what happens. And this is, again, an empirical observation. We have our empirical neural space, N1, N2, and N3. At any time, we have our window, our time window of size delta, and we can measure the activity of each one of the neurons. So at every time, at every one of these discrete times, the activity of the whole population is just represented by one point in this neural space. This point has the coordinates N1, N2, and N3, has the coordinates activity, firing rate of each one of the neurons that I'm recording from. And now we allow this window to move through time, and we follow these points as a function of time. So now we have a trajectory of activities in this neural space. And what is seen empirically is that these trajectories are not exploring the full dimensionality of the neural space, but that they are constrained to low-dimensional structures. And these low-dimensional structures are the manifolds that I want to talk about. So these neural manifolds are the low-dimensional structures to which the neural activity is confined. These neural manifolds could be flat, that is to say linear, or could be non-linear, like the blue one that I'm showing you here. If it's flat, that's the simplest case, because if it's flat, then we can describe this with a basis, like in this case U1 and U2. Each one of these vectors is a direction in neural space, is what I called a neural mode before. It's a particular combination or a particular pattern of activity, and that's U1 and U2. And in that case, we will be doing a linear dimensionality reduction to find this hyperplane, essentially, within the empirical neural space. Or it could be that it is a curved manifold, and in which case, of course, locally we can perform a linear approximation, but the basis, the vectors that allow me to do that linear approximation would vary depending on where we are within the manifold, and therefore the description will be more complicated. So this question of whether the manifolds are linear and nonlinear is a very, and how to find the nonlinear manifolds is a very active area of research in this particular subfield. So what does it mean to have linear or nonlinear manifolds? So let's look at this simple ring. So take I don't know, a little cable and just fold it onto itself. So this ring has an intrinsic dimension of 1. Now, if it's flat, I can put it on the surface of the table, and I can see it within a flat subspace that has dimension 2. So the intrinsic dimension is 1, but if the ring is totally flat, I can go to a 2-dimensional space. Here I have principal component 1, 2, and 3. I could neglect 3, and I could talk about a flat subspace of dimension 2 that allows me to see this ring, although it's a nonlinear manifold whose intrinsic dimension is 1. But now imagine that you have this ring, and now you pinch it in a particular direction, and you pinch it in the 3rd dimension. So now you need a 3rd dimension, although the intrinsic dimension has not changed, it's still 1. And now if you start pinching it, if you're in a 100-dimensional neural space and you start pinching it in all the possible dimensions in different places, then you have to increase the dimensionality of the flat space that would allow you to see this ring. So in some sense, there is an embedding flat dimension, which is the smallest Euclidean space, the smallest flat subspace that I need to consider in order to be able to see the nonlinear manifold. But the dimensionality of that embedding dimension, which is a flat dimension, is going to depend on the degree of nonlinearity. The more pinches I make, the more nonlinear I make this structure, then this embedding dimension, the flat dimension, would grow, although the intrinsic dimension is always 1. It's the same thing if I'm walking on the surface of a sphere. If I'm limited to the surface of a sphere, the intrinsic dimension of my manifold is 2. The flat dimension is 3. That's the dimension of the minimum of the smallest flat space within which I could see the nonlinear manifold. And we had a workshop at the Computational Neuroscience meeting in 2019 on these issues, and both Sara Ostojic at the École Normale in Paris and Ilafite at MIT made very beautiful examples that made this point very clear. So let me give you yet another way of thinking about this. So we are recording from these simple neurons, and 1 and 2 and 3. A few of them, we have this very low dimensional empirical neural space, very low dimensional as compared to the true neural space, which would be millions of neurons, or 1 billion neurons in this case. Let's say we can consider just a flat manifold that contains most of the dynamics. Here the points are getting bigger just to show you in which direction time flows. And because this manifold is a hyperplane, is flat, we can just consider the bases U1 and U2 that we can find using principal component analysis or factor analysis. This will give us 2 eigenvectors that are unidimensional and orthogonal to each other, and that provide a basis. Every point in this hyperplane can be written as a linear combination of these 2 vectors. Therefore, instead of projecting the dynamics on the axis given by the neurons, neuron 1, neuron 2, neuron 3, I could project the dynamics on these 2 directions. I could project the dynamics on the direction of U1, and I would get a curve which is latent variable 1, which is the degree of activation of that neural mode as a function of time, and the same for U2. So I have 2 alternative descriptions. One description is the neural activities that tell me as a function of time how is the activity of neuron 1, neuron 2, and neuron 3 evolving. And the other description is as a function of time how are these latent variables evolving. And the interesting thing is that the latent variables are a generative model. This low dimensional representation gives me a generative model for the activity of the full neural population. And the coefficients of the linear combination that allows me to go from latent variables to actual neural activations, neural variables, are exactly these vectors U1 and U2. So these are the 3 components of the vector U1, written now as a first column, and the 3 components of the vector U2. These vectors are, of course, have 3 components because they are vectors in a 3-dimensional neural space in this simple example, and we'll show how to do it in general in the next part of my talk that we're moving into. But the interesting point is that I can reconstruct the neural activities from the latent activities. So in this sense, the latent activities, the latent variables, are the building blocks of the population activity. And if I know the time evolution in this low dimensional representation provided by the neural manifold, I can also know the time evolution in the large dimensional neural space. So that's the point that we made with Juan Gallego and Matthew Perich in a paper, in a kind of mini review that we wrote in Neuron in 2017, that we discussed a little bit earlier as a journal club with some of your graduate and undergraduate students. And we talked about the neural modes, the neural manifold, and we talked about latent dynamics, which is a time-dependent activation of these neural modes. And we talked about the fact that these are the building blocks, the generators of neural activity and therefore of behavior. And the point that we made in that paper, although that paper was mostly a review of the observation of neural manifolds in motor cortex, was that these neural manifolds or neural modes, low-dimensional representations, have been observed in other brain areas, like frontal and prefrontal cortex, parietal cortex, visual, auditory, and olfactory cortices. So not only motor, but in sensory cortices and also in higher order cortices like frontal, prefrontal, and parietal cortex, more related to functions,
Sara?
like decision making. And let me say one thing, and I'll take your questions. So this is just, a list that as of 2017, but of course it can be updated with much work that has happened since then. Yes.
Okay, we have 3 more questions. I think they're relatively quick. So Yunchan Zhang is asking you, how would the size of the time window for binning affect the shape and the dimensionality of the manifolds? Is there such a thing as an optimal time window for looking at neural dynamics?
That's very interesting. Of course, the choice of the time window allows us to move from a spike timing representation, if you choose a window from 1 millisecond, to a very coarse grain fine grain representation, like in the case I showed you earlier of 300 milliseconds. The resolution of the time window will simply allow you to change the number of points that describe the trajectory. So let me go back to this slide. If I increase my time resolution, I will have more points along this continuous curve. But if this continuous curve lies on a low-dimensional manifold, that should not change. Increasing the time resolution would not change that fact.
Okay. Have you given any thought to what kind of neural dynamics would generate manifolds of this particular type?
That is very, very interesting, and that's an area of very active research. So beautiful work has come recently from the lab of Søren Osterjic in Paris, where he relates the rank of the connectivity matrix of the neural ensemble to the dimensionality of the dynamics. And he has shown that if, for instance, if you start with a recurrent network that is randomly connected, it will have full rank. And if you just let it run its own dynamics, it will be full dimension. But if you now train this network to do something, like for instance, reproduce the kinematics in the simple movement of the cursor in the screen in the XY plane, you train the recurrent network to do that. Now the trained network has a connectivity, the changes in weights due to the training are a low rank matrix, and that low rank is associated with the dimensionality of the corresponding dynamics. So it's the reason why these low dimensional dynamics arise really has to do with the connectivity of the matrix.
Mm-hmm. 2 more, and then we can go again. What happens if you have like a sequence of motions? So like a sequence of motifs A, B, C. How would the manifold of the sequence relate to the manifold of the individual components? If I'm understanding the question correctly.
I would say that the manifold of the sequence is a union of the manifolds. If you're going to use these motifs in other context, I mean, if you are only going to use these motifs on the sequence, then you have a representation for the sequence itself. But if you are going to use these motifs in a different order or in different contexts for different tasks, then what you are going to have is a manifold for each one of the motifs, and then the sequence would be a particular union of these manifold of motifs.
And the very last thing, how does the dimensionality of the manifold relate to the complexity of the task?
That's a
like if you know something about the task, can you anticipate what the manifold dimensionality should be?
That is a very, very interesting question, and our colleague Surya Ganguly at Stanford has done beautiful work on that. He has a beautiful paper in which he relates task complexity to the dimensionality of the corresponding manifold of the task-specific manifold. I've shown you here a very, very simple task in which I can get away with a two-dimensional manifold. But if we let the monkey move freely in a cage, and where he's, scratching himself or reaching or hanging from the ceiling and strutting around and doing different things, and we look at the dimensionality as if this was a single task without segmenting, then we find the dimensions that are not 2, but they are 15 or 20. We don't find dimensionalities that get close to 100 that would say, oh, we're being limited by the number of neurons that we are recording from. And this is to me an indication that the dynamics truly, fundamentally lives in a low-dimensional manifold, and that it is a consequence of a particular wiring of the network, the wiring structure of the network, the connectivity.
Okay, I think we're ready to continue.
Okay, so let me go back to where we were. And let me move, then I think I told you everything. Yes, I told you everything I wanted to tell you in general. And so I want to give you a particular example of analysis. And the analysis is going to be all linear. So I'm going to make, there are 2 things that I want you to remember from my talk. One of them is that these population dynamics are confined to low-dimensional structures. And the second one that I want to convey to you is that whenever we are confronted with the dynamics of a very complex, high-dimensional interconnected system or any such complex behavior, we should always start with the linear approach. We should see how far the linear approach takes us, how much we can learn from the linear approach. And then we should try, of course, to break it. And the way that it breaks gives us some clue, I think, hopefully, on how to go nonlinear. Because once we go nonlinear, the universe of approaches and methods and models is essentially infinite. And it's not always easy to have an intuition of how to introduce nonlinearities into your model. So always go linear, and I'm going to show you a case. Always start linear, and I'm going to show you a case in which the linear approach is unreasonably effective. So I go back to this task that I showed you before. So this task is a task that the monkey has learned very well and repeats in a very stereotyped manner. It's like if you see, I don't know, Nadal serving, he always serves in exactly the same way. So here I show you data from day 1. This is the trajectories in all 8 targets, again color-coded. And if you take a given target, like the one that is to the upper right, and you look at the x and y components of velocities within the reach, you see also that the x component and the y component of velocity is very well correlated with itself. Now the monkey is doing other things, and a year later comes back to the lab and does his task. He has done it a couple of times in between. But we measure again, we look at the trajectories, they are pretty much the same, and the x and y components of velocity are also very correlated. And then more than 2 years later, we test for this again, and we find exactly the same stability in behavior. The trajectories are the same. And if you look at the correlations of hand velocity, both in the x and y direction, these blobs correspond to the times when the monkey has been in the lab doing that particular task. But this is over a period of almost 800 days. And you look at the histogram of those correlations, you see that's very strongly clustered around 1. So this is just to tell you that the behavior itself is very stereotyped, both in the trajectories that are described and in the velocity profiles that are used to span those trajectories. And so we had an hypothesis, which was there has to be stable neural dynamics underlying this stable behavior. That if the subject consistently performs the same behavior over days, months, and even years, then there has to be latent dynamics that is also stable. But in order to verify this hypothesis, we had a problem, which is that over these 2 years that the electrode array has been implanted, we are not always recording from the same neurons. Some electrodes, scar tissue grows around them and we lose that signal. The electrode array itself goes a little bit deeper into the brain, and suddenly we capture new neurons that are not the same as we were capturing before. So on day 1, we are recording from a subset of neurons, and on day n, we're recording of a subset of neurons which could be entirely different, not entirely different, but have small overlap, like a 20% or 10% overlap with the neurons that we're recording on day one. And so how are we going to verify the hypothesis of stable neural behavior when we are not recording from the same neurons? So what we have is a large population activity, but the ones that were being recorded, the empirical neural space that we are analyzing is different on different days. So we have to find a way of compensating for this difference in empirical neural spaces. So this is the guiding slide over the next few slides that I'll show you, what's left of my talk. So this is the belief is that there is a huge neural space, let's say the million neurons that are being modulated by this task. And in this true neural space, there is a true neural manifold, the mother manifold that is there. And in this manifold, there is true neural dynamics that is very stable, is being preserved. Now, on day one, we come and we measure a few neurons. So there is a projection of the true neural space in a particular empirical neural space on day one, which is going to be different in principle. Maybe some neurons overlap, but it's going to be different in principle from the embedding space on day n. So there are two projections to an empirical neural space on day one and day n. Projections are linear, but they are different. Now, in this neural space, we do a principal component analysis. We find an empirical neural manifold, and in this empirical neural manifold, you observe latent dynamics. Again, because we're doing principal component analysis, that's a linear dimensionality reduction on day one and another linear dimensionality reduction of day n. Well, this dynamics doesn't look at all like these dynamics, but we ask ourselves, since both the projection from the true neural space to the empirical neural space is linear, and the projection that we use to do the dimensionality reduction, principal component analysis, is also a linear operation, is there a linear method that would allow us to align the dynamics, the latent dynamics in day one and the latent dynamics in day n, so that we can say that they are actually the same dynamics, just seen from different points of view? And the answer is yes. So let me just walk you through the math. So we start with this data matrix. Again, each column represents a time. This is the duration of the experiment. Each row represents a neuron. And the neurons that we have taken is actually the union of the neurons recorded on day n, let's say, and the neurons recording on day m, or the neurons recording along all days. And so we have a data matrix of dimension d by t, where the ambient dimension is the cardinality of the union set of the neurons recording on day n on day m. And so we have a matrix for day n and a matrix for day m. And in each one of these matrices, we do a singular value decomposition. The U matrix gives us a basis for the neural space on day n, and this U matrix gives us a basis for the neural space on day m. The column vectors of these matrices are ordered according to the eigenvalues that appear in the sigma matrix, so we can do principal component analysis. We can just say, instead of having capital D dimensions in the neural space, we are going to only keep lower d dimensions by going into the matrix U, both UN and UM, that were the dimension d by d, and just keeping the leading columns, which is the leading eigenvectors corresponding to this matrix. So now we have lowercase d, a hyperplane that is spanned by the columns of u and tilde, and another hyperplane that is spanned by the columns of u and tilde. And these are day-specific neural modes, and we are talking about a flat dimension, right? Where I'm just doing everything linearly, principal component analysis. And I'm going to keep lowercase d big enough so that if there is a nonlinear manifold, I'm not cutting it off. I'm hoping it's embedded in this flat dimension. There is an intrinsic nonlinear manifold embedded, of intrinsic dimension, embedded in this hyperplane that has a flat dimension D. So we project all the data on these hyperplanes using now the rows of this matrix U, the eigenvectors as rows, and we get the latent variables, which are now of dimension lowercase d by t instead of capital D by t. So this is just the projection onto the corresponding hyperplane, both on day n and day m. And now we do canonical correlation analysis, which is the linear method that will allow us to align the dynamics. If we were looking at the unaligned dynamics, we would have to look at the cross product, the scalar product between the latent activity at AM and the latent activity at AM, and we're not going to get much of a correlation because these are very different latent dynamics. So what we do is we do a QR decomposition of the dynamics on day N. Now I'm taking it as a transpose, so now the columns are the dynamics, and the same thing on day M. And the Qs are going to give me orthogonal representations of those dynamics. The Rs are the triangular mixing matrices. And now what we do, instead of projecting L onto L, or L transpose onto L, we project Q transpose onto Q. And we do, again, a singular value decomposition, where now U gives us a basis for day N, and V gives us a basis for day M. So an S gives us the canonical correlation sorted from largest to smaller, and these are going to quantify the similarity in the aligned data dynamics. So we use, yes?
Sara, we have more questions. So Arjit was wondering to which degree you think these manifolds are in some way aligned across animals. I know that this is
I will comment on that. Yes, they are aligned. We can not only align them across days for a given monkey, but actually Matt Perich, who is younger than I am and therefore braver, decided to check whether we could align them across animals. And I thought this was the craziest thing I had ever heard, that it was never going to work. But he ignored me and did it, and it worked. And so Matt is preparing a paper on that. We can align the manifold for this very simple task, which is made in a very stereotypical manner by all monkeys that we have trained. We have been able to align across manifolds in 4 different monkeys.
Excellent. And Vishwa has another question. He wonders if it's possible to test whether the true neural manifold is fixed across this time scale, or if the manifold geometry is preserved, but the participating neurons change their tuning over this time. So if there would be a systematic rotation of the axis or the drift of the axis across time, would you be able to tell?
Yeah, let me just go 2 slides further and comment on that, and you ask again if I haven't answered the question. So we have a matrix U for day N and a matrix V for day M, and I'm going to use these matrices to do a linear transformation on the latent spaces. So I'm going to use U to construct the matrix M and V to construct the matrix M, and use the inverse of the triangular matrices, so that now I can effect a linear transformation in the corresponding latent spaces. And we learned this method from the beautiful paper of Bach and Jordan, and the proposed canonical correlations analysis in a completely different context. And this is what we get. And this is really the punchline here. So we took our data on day 1. So what I've done here is I'm showing you in a 3-dimensional space, because now I'm following the whole dynamics. I have average over all the trials for a given target. So I have 8 trajectories color-coded by target on day 1. And I have, again, 8 trajectories color-coded by target on day 32. These neural modes are not the same as these neural modes, because the empirical neural space on which I have projected on day 1 is different from the empirical space on which I have projected on day 2. So these are these are the matrices U and V. So these are the eigenvectors that I'm using now, right? So these are the neural modes. Nothing happens so far, but then I align the dynamics. And when I align the dynamics, I do a linear transformation on this latent space and a linear transformation independently on this latent space. What I find, to me, was truly amazing. First of all, they look very similar. So I have really aligned the dynamics. I have kind of 1 petal per target. So this describes the dynamics of going out to the target and coming back to center. And moreover, the petals are organized the way they were in our very simple analysis and the Krishna Shenoy analysis, where we didn't take into the dynamics, we didn't fine grain into the dynamics at all. Again, we go yellow, orange, green, aqua, yellow, orange, green, aqua, yellow, orange, green, aqua. This representation again captures the organization of the targets in real space. So we believe that these aligned representations that we have obtained by doing linear transformations in each one of the low dimensional flat manifolds in each one of the corresponding neural spaces are really a reflection of the true object, which is the true manifold. Now, let me go back to the last question, which I didn't answer. The last question was, could there have been a change in the mother manifold that I am not detecting? And the answer is yes. If there had been a linear change in that manifold, this method would compensate for it and I would not be able to detect it. So there is that caveat. So this is consistent with the idea that the manifold is stable, that the true manifold in the full neural space is stable, and that the linear and the dynamics, the latent dynamics within that manifold are stable, except for the possibility of linear transformations. And just to be more quantitative, this is in gray, I'm showing you the alignment on the same day. In orange, I'm showing you alignment across sessions. This is the days between sessions. If we don't do this correction, and then the red is after we have aligned. So after we have aligned, we get such good correlations, same as we get if we compare the early session with the later session within a given recording day. And we have verified that not only for that monkey that I just showed you over 30 days, but also over the 700-day monkey and over the 70-day monkey and over the 500-day monkey. So this is more than 1 year of data, this is more than 2 years of data, and this is more on the scale of months. So I'll stop there. So this is really the picture. Well, let me just show you this. The other reason why we know that this alignment method works is because we use these latent dynamics as inputs to the coders, as inputs that, as a very simple linear decoder that takes the activity, the recorded activity in primary motor cortex, and produces an XY trajectory on the screen of the computer. And instead of using as input the full recording, we use as input the latent variables. Now, the latent variables on day N are not aligned to the latent variables on day 1. So if we train the decoder on day 1, the performance of that decoder amazingly deteriorates, rapidly deteriorates as days pass. As you see, by the time you have over a month, the activity that you're recording is so uncorrelated with the activity that you are recording on day 1 that this linear decoder no longer works. So you can say, well, let me retrain the linear decoder every day at the beginning of the sessions. Let me take data, retrain the linear decoder. It's just a linear decoder. What's the problem? The problem is that if you want a human to use this or the monkey to use it, when you change the decoder, you have to adjust yourself again to the decoder. You have to adjust to providing the correct activity to the new decoder. So instead of doing that, what we do is an alignment. We take the data on day N and we align it with the data on day 1 as a pre-processing before giving it as input to the decoder. And we give it, when we do that, the performance of the decoder is as well, again, as the gray, which is decoder designed or established the parameters of the decoders in early part of the session, and then evaluating the performance of the decoder in later parts of the session. So this kind of gives us a sanity check that what we're doing makes sense. And this will be my last slide. I just want to leave you with this picture of a true neural space with a true neural manifold, with a true latent dynamics that we can explore by projecting it into an empirically accessible neural space, determined, whose dimensionality is determined by the number of neurons we can record. And within that space, we can do a linear dimensionality reduction that has to be ample enough to contain potential nonlinear manifolds that are embedded in that hyperplane. And why can we trust this to be good reflections of the full representation? Well, this possibility of aligning across days and even aligning across animals is what gives us confidence on the existence of this abstract object that we cannot really map directly. And I will end there.
Thank you. That was absolutely excellent. We have already a few questions in the queue, but feel free to add more and upvote the ones that you like best. So the first one comes from Artem. He's wondering if there's a way to meaningfully quantify the properties of manifolds. For instance, looking at the topological characteristics like Betty numbers and things like that. And would that tell us something about the nature of the task or about behavior?
So as long as we stay with this idea of the flat manifolds, which potentially contain a lower dimensional nonlinear manifold. In the flat manifolds, it's very easy to characterize because we go to the center of mass and we compute its distance to the origin, so we know how far away from the origin we are. And the eigenvectors that give us a base for the manifold define the orientation of that manifold. So if I know the orientation of the hyperplane and I know how far from the origin I am, I know everything that I need to do about the hyperplane. Now, even that hyperplane is not fully explored. I mean, the data is confined to that hyperplane, but it's not fully explored. And this is the notion of the convex hull of the data, or what my colleague Byron Yu in Pittsburgh calls the repertoire, which is some kind of a curve, so that all the points that are really visited would be within. And you know that this is, even within the hyperplane, this is the region that is being explored by the system. This contains the states that are being explored by the system. And that, of course, is much harder to characterize, that border, because that would be probably a very wiggly curve, harder to find out. But I expect all these features to be task-specific. Mm-hmm. That answers the question.
Right. So Pavel's asking, manifolds and latent dynamics seem like a very, very elegant characterizations of neural recordings. Can you speculate how does the brain have access to this information?
Yes. So I can speculate mostly motor cortex, which is what I know. So motor cortex is just 2 synapses away from muscles. These neurons in primary motor cortex in M1 project onto neurons in the spinal cord, motor neurons in the spinal cord, with themselves control muscle activity. So if I am a neuron on spinal cord, I have to wire myself. I'm a readout neuron, right? So what simpler job could I have than to be able to do a linear readout within a low-dimensional manifold? So I just have to wire myself to be able to read the latent variables, which are only a few of them. So a few of these motor neurons, each one of them tuned to read a given latent variable, can give me a full description of the dynamics in the manifold. So here it goes a very provocative speculation. When we look at early stages of sensory processing, I expect to find very nonlinear manifolds, because the activity is a representation of a world in which nonlinearities are very important. For instance, in vision, edges are very important. If I can make a caricature of you just with a few on showing the edges, then people will be able to recognize you, right? So these nonlinearities are crucial in the actual natural images, and therefore in the early stages of sensory processing. But I believe as you go up and then down to motor cortex, the brain linearizes the representations in the same way that it does in a deep neural network, in a deep artificial layered neural network. And it linearizes the representation, and it's always surprised me how we get away with linear methods in analyzing motor cortex. When I do sophisticated non-linear methods, the decoders improve by 2%. They don't improve by 10% or 20%. Which means that my linear representation was a very good representation of what was going on. And I've always bothered me, why would motor cortex be so linear? And I said, well, it makes a lot of sense, because I make the job of the motor neuron, who has to read that output and send instructions to the muscle, much easier. So it's purely speculative, very provocative. If you quote me on that, please quote me. I
very cool.
Very carefully between what I know and when I'm speculating. And in this case, I'm just speculating.
Great. Benoit has a really cool question that I think would be nice to address. So he's finding it very surprising that the manifolds are so remarkably stable given plasticity and learning. Is it that these monkeys don't particularly do anything other than reaching to various places?
That goes to my point about practicing. These are not these are tasks that you acquire, but then you keep on practicing. And by practicing, you reinforce the connectivity, the particular pattern of connectivity that gives rise to this manifold. And this is where the memory is. The memory really is in the connectivity. What we don't have yet is a good theory of how the network connectivity results in this low-dimensional dynamics. But what we reinforce is the network connectivity when we practice. And that, as a consequence of that, there is a stability in the manifold and a stability in the dynamics within the manifold, the intrinsic dynamics within the manifold.
Okay. We have a lot more questions. I don't know in which order to go. Maybe a technical one. So Julie Yu is wondering, since you're doing this, like finding linear projections to do the aligning in consecutive days, is that like a smooth progression across days? So if you're looking at a couple of alignment matrices in close by in time, would those be more similar than further apart?
See that again? Could you go through it again? I'm not sure I get the question. Or maybe I can read it myself. Hold on.
So it's about aligning manifold using linear transformations. Are these transformation, like your alignments, somewhat smooth with respect to consecutive days or consecutive measurements?
Oh, I see the question. How do these matrices vary as a function of time? I haven't checked that. That's a very good question. I would expect them to vary by small steps because the number, as we go from a day to the next, is only a few number of neurons that are different. But if you take days like 20 days apart, then there will be less overlap between the number of neurons. But I haven't checked that. That's a very good point.
Maybe we'll take a very last one and then we will wrap up. We're already 1 hour and 15 in. So Ben is wondering, how does the learning a new manifold work? So do you need to sort of ping the system in a very different region of the neural space and then start building a representation there?
Yeah. So there's beautiful experiments coming from the lab of Aaron Batista in P, where he did the following. So he worked with the decoder. So the decoder is looking at specific directions in neural space, right? Every unit in the decoder is looking at a linear combination of the recording neurons. So that's a vector in neural space. So the decoder lives in a subspace in neural space, in a hyperplane in neural space. If I change the decoder by reorienting those vectors within the hyperplane, the monkey learns a new decoder very rapidly. But if I change the decoder by taking one of those vectors and giving it a component out of that hyperplane, now I have a component of a neural mode that was not part of the task before. I have to move in a direction of it. And that learning is very slow, takes days instead of hours, and it's as slow as it was to learn the decoder to begin with.
Mm-hmm.
Which means that once I have acquired some neural modes, I have acquired a task, I have acquired neural modes, I can reuse those neural modes very easily. But if I have to learn a new neural mode, well, that is as difficult as it was to learn those neural modes to begin with, which also is a very nice idea to understand why having learned something can facilitate the learning of something else. Because if there is some commonality in the directions of these two hyperplanes, of these two manifolds, so that some of the neural modes can be adapted to the new task or almost identically used to the new task, it will be much easier to learn a new task than if you had to learn it by learning not regions but directions in neural space that are completely different from directions in neural space that you have learned before. I hope that answers the question.
No, that was absolutely great. I think we're ready to wrap it up. Let's thank Sara again for a very excellent talk. And as a reminder, this is the very last talk of the series. So we're ending up this semester, and we'll be back in the fall. Hope to see you all of you there again. And thank you again.
A final word. First of all, I would like to thank you very much for the invitation. I've enjoyed my day so far, and I plan to keep on enjoying the rest of the day as I talk to various people. But I wanted to say a word about the graduate student situation at NYU. I wanted to say, it's not my place to comment on an institution that is not my institution, and I'm a guest. But I want to say that we have all of us who are faculty or postdocs, we have all been graduate students. We know how tight the money was. I can't imagine how tight the money is in a place like New York, which is such a expensive place to live. But I want to say that we really need to rethink how we recognize the work of graduate students. Because if they are TAs, they are really supporting the teaching mission of the university. In a way, it would not be possible without them. If they are research assistants, then they are helping us carry out experiments that allow us to publish and publish the results of work that we've been funded for, and then renew the funding so that we can continue the work. So either when you consider the educational mission or the research mission of the university, the graduate students are a fundamental part of it, and they have to be recognized as such. They shouldn't be treated as cheap labor. They shouldn't be treated as second-class citizens. They should be given a living wage. They should not worry about their health insurance. And I wish the graduate students at NYU the very best in their quest for getting to those conditions. Thank you.
Thank you. Thank you again. Bye, everyone. Thanks.